depending on where you find yourself in the world today. I'm excited to be with you again today, and I see a lot of familiar faces from our previous calls, but I also know we expect a lot of new people. I guess I've been tracking with Bob and Sherry and Bill to see kind of how things went after our meeting last Saturday. And it sounds like we're really lighting up. Some people have really engaged to take advantage of the offers that were made on Saturday. So I'm excited. And so what we thought the best use of our time today would be is, since the introduction videos that we have all taught, the videos that we have on the Share app, they all talk about Glycocalyx, and they talk about microvascular. And I know previously a few weeks ago, we had some in-depth talks about that, but we have a lot of new doctors on the call today, or that will be wanting to view this presentation again. So what I want to do today is have Dr. Vink go back and talk about the journey of the discovery of the importance of microvascular health, number one, and number two, explain the endothelial glycocalyx, and then show the scientific research and data that got us to the point that we're at today. And I'm going to turn the time over to Hans, but before I do, I don't think I've ever shared this with the group, and I just wanted to let you know, one of the other things that Hans has done for many years, in addition to being a research scientist, is he's also been a professor and has taught physiology at the medical school at the University of Maastricht. And so he typically teaches physiology to the doctors, and he does a lot of teaching with the doctors that at the end of the rotation of their residency. And so they bring all of their hard physiology questions, and they want to know what's happening here, what's happening here, why did this drug do this, why did this happen? And so what I've come to learn with Hans is he has an incredible knowledge of the human body and all the things that happen, and how all these different diseases and condition impact our health. And so with that, I'm going to turn the time over to Hans, and we're going to take about 45 minutes, I think, for him to go through that. And then we're going to have a quick other guest come on for a minute, and then we're going to try to reserve 10 or 15 minutes at the end for specific questions. So if you have questions you don't understand, we can answer that for you. So with that, Hans, I'm going to turn it over to you, buddy.
Well, thank you, Bob. So as Bob mentioned, what I would like to do with you today, again, take a little bit deeper dive into the research that we did to look at the microvasculature, and then finding out that we needed to know more, better understanding, because things didn't look right. And then how we came to study Glycocalyx, how to visualize it, and then all the important functions that are associated with not only microvasculature, but also the important role of Glycocalyx. So what happens with your vascular system if your Glycocalyx gets damaged? So I want to spend some time on that with you today, and not as much on the GlycoCheck system itself or on the therapeutic, just to give you a better foundation on the science. So let me switch cameras first and start sharing. All right, and then show. There we go. I hope you can see my screen now. So take it from the start again. So we know that if you look at your vascular system, that the large vessels that take the blood from the heart to your extremities and to your organs, and also collect the blood from your organs and bring it back again to the heart, that those vessels only make up 1% of your vascular system that is in touch with your tissue cells. So if you take a microscope and look more closely, you see that anywhere where there's a tissue cell, there's also a microscopic small blood vessel very close to that cell to take the nutrients and the oxygen to those cells. So the way that all the nutrients travel to the tissue cells is by simple diffusion, and that only works at very short distances from each tissue cell. So you need many, many small vessels, one vessel close to each tissue cell. So more than 99% of all the contact between your vascular system and your tissue cells is in those microscopic small vessels that we call the capillaries. And when we look at one of those individual capillaries, and look in living tissue, and we look at how red blood cells are flowing through those individual capillary blood vessels, well, we realize when we look very closely that these individual red blood cells never touch the inside of the vessel wall. So these tubes are tiny. They're so small that one single red blood cell actually doesn't fit. So that individual red blood cell needs to be deformed. They're easily deformable. They're very soft. And then they fit into this little tube of a capillary blood vessel. Then again, they never touch the wall. And now we know that in between the surface of the wall of the vessel and where the blood is actually flowing, there's this invisible, transparent, protective compartment that we call glycocalyx. So if we zoom in a little bit more, we got these kinds of images with a different kind of microscope called an electron microscope. Again, this is a cross-section of one of those tiny capillaries. And if you look at the higher magnification on the right-hand side, so here's the wall. So the wall consists of only one cell called the endothelial cell, which is the actual wall of those tiny capillaries. And those are very thin, only 1/10 or 2/10 of a micron. And on the surface of that endothelial cell, you see all these complex three-dimensional structures, which are long sugar chains called polysaccharides, which are produced by the endothelium, anchored into the membrane and sticking out into the lumen of the blood vessel. And here on top is where the red cells are flowing. So you can appreciate, relative to the size of the endothelial cell itself, these bush-like structures which make up glycocalyx are huge. They're about 5 to 10 times or more the size of a single endothelial cell. So the glycocalyx extends microns into the lumen of these tiny vessels and occupy a lot of volume. So one of the first aspects and implications of this glycocalyx bush that I would like to discuss with you is the volume aspect. If we go back again to the previous slide, this one, just to give you an understanding of the volume aspects, this vessel, a small capillary, the diameter of the capillary is about 5 microns. And the volume of a capillary blood vessel goes with the power 2 of the diameter. So a diameter of 5 microns means that the anatomical volume would be 5 times 5 is 25. However, the actual volume where blood is flowing is much smaller, because on each side there's 1 micron space occupied by this transparent glycocalyx gel. So the actual diameter where the red cells and the blood plasma are flowing is only 3 microns in diameter. So the volume of the blood flowing in this capillary is only 3 times 3 is 9. So only 9 out of 25 of the total volume is occupied by the red blood cells. This means that 16 out of 25 is occupied by glycocalyx, occupying a lot of space on the inside of those vessels. So 2/3 of the volume would be glycocalyx. Only 1/3 of the volume is blood volume under resting conditions. So again, the reason that I mentioned this is that actually triggered the questions that led to the discovery of glycocalyx. So this video that I'm showing here right now is an old video, and the types of video that we would start to collect when we started looking at the microvasculature with big microscopes, and people started looking at those kinds of tissues with these microscopes. in the 1970s, 1980s. And people taking these kinds of videos, they were actually puzzled by the fact that if you look at the number of red blood cells in these tiny capillaries, the red cell volume fraction appeared to be less than 5 to 10% instead of 50%, what you would expect if normal blood would be occupying all the available volume in these small capillaries. So this triggered that question, why is the blood volume in these capillaries so low? And I had the privilege, when I started as a young student, to work many years with this person, Brian Dooling, in Charlottesville, Virginia. And he already at that time, when I started as a young student, made those observations and started speculating that maybe something else, and we didn't know about Glycocalyx at that time, maybe something else was also inside these tiny capillaries, occupying a lot of volume, and that causing that the red cell content would be low because a lot of the volume is already occupied by Glycocalyx gel. And another observation that he made is that although most of the time under resting conditions, like on the left, only few red cells were seen inside these capillaries, and a lot of the volume was occupied by something else. But as soon as you start exercising or do something else, which asks for more blood volume and more nutrients to be delivered to your tissue cells, then suddenly, within a second, many more capillaries would be perfused with red blood cells, and many more red cells would fit inside these capillaries. So not only was it hypothesized that under resting conditions would be something inside these capillaries occupying a lot of volume, but also that whatever that was, that its volume would be very dynamic. So it would go much bigger and smaller depending on demand of the tissue cells. So controlling red cell volume and blood volume in tissues upon demand. So one of the first questions that needed to be addressed is whether the blood itself flowing through these capillaries under resting conditions, if the red cell concentration in the blood itself was still a normal 50%, as what you would expect based on larger vessels. So on the left, you see a larger vessel, an artery, taking the blood to the microvasculature. And if you take a blood sample from a large blood vessel and spin it down in the centrifuge, you'll see that 50% of the volume of a blood sample is red blood cells. While if you take a picture like this from the microvasculature and you count the number of red cells and you look at the estimated volume of those microvessels, now those estimates give you estimates of red cell concentration as low as 7%. So many people said, well, the blood must be diluted. There's more blood plasma, the clear liquid, compared to red cells, and that might be because maybe the angle of the small vessels relative to the large vessels makes it only fluid goes in and no red cells. So first we needed to test, if you take a blood sample from an individual capillary under resting conditions, is the red cell concentration still 50%, or is the blood diluted because of the angle of the small vessels with the larger vessels? So Brian Dooling actually designed these very tiny small pipettes. So those are glass micropipettes. And so he pulled with very small tips on the end and then sharpened them. So a very sharp tip with only a few microns in diameter. And with these very small micropipettes, he could actually puncture individual capillary blood vessels to collect the blood flowing through those small capillaries. And if you look at the bottom two graphs, they show on the vertical axis the red blood cell concentration in the blood collected from vessels with different diameters. So from left to right, he started out with larger vessels, and as expected, the red cell concentration in the larger vessels was 50%. Then when he moved more towards the smaller vessels up to the capillaries on this end, the blood collecting from the capillaries was still a 50% red cell concentration. And then he moved on to the exit part of the capillaries, and to the small collecting blood vessels, so from small again to large. But all the different diameters of blood vessels, they all had the same red blood cell concentration. So the blood was not diluted flowing into the microvasculature. So something else must have been affecting the volume available for the red cells in those capillaries. So the hypothesis that he and another person called Bruce Klitzman came up already in the late 1970s, that maybe if you look at the capillary schematically, that on the inside of this tube, there would be what he called an hypothetical plasma layer. So a layer of transparent clear liquid that actually was moving very slowly, not moving at all. And at that time, well, there was a lot of skepticism about this hypothesis because 9 out of 10 people would consider these small blood vessels just glass tubes. And there was no reason that in the glass tube, any liquid or fluid would stop flowing near the edge, near close to the wall. So he was ridiculed for many years, not finding any evidence for another compartment that holds the plasma immobilized inside a vessel occupying a lot of volume. Well, a next important step to get more evidence that something else might sit inside the capillaries and limiting the red cell content was when he started doing experiments with a person called Claude Desjardins. Claude Desjardins had worked on different topics with a lot of enzymes, studying the matrices in tissues. So he worked with Brian to test different enzymes that might break down whatever was keeping the fluid immobilized near the vessel wall. So again, they took these small micropipettes, they injected different enzymes that break down different polysaccharides into small capillary networks, and then comparing the red cell content with networks that did not see the enzyme. So again, on the lower left is a normal baseline condition of the microvasculature, again, showing only a small part of the volume inside can be used for blood flow. Then using the enzymes, you could actually flush out whatever was occupying that space, and then show that after you remove that, still mysterious transparent compartment out of the capillary, then suddenly all of the volume of the capillaries would be accessible to red blood cells, and the red cell content could, within an instant, could increase 3 or 4-fold, and many more capillaries would show up, showing a lot of red cell inside. So that was about the time that I started visiting Brian in Charlottesville. So my first objective was to find a way to visualize whatever was inside these capillaries that was modulating red cell content. And one of the ideas we had was to use fluorescent dyes to label different plasma proteins to see if any of those proteins were part of this mysterious compartment inside these vessels, and we could see proteins stick to the wall. So the dark gray area inside this vessel represents the dye-labeled proteins that are infused, and then the dotted circle is the area that I turn on a very bright light to excite the fluorescent dye to see if the dye would stick to the wall, and find evidence of a glycocalyx on the inside of the vessel wall. And I tried doing this for more than 2 years and couldn't see any of the proteins stick to the wall. And then at some point I realized, I turn on the light, I don't see anything, turn off the light, and again look at the capillary. And then I realized that exactly at the area that I had been looking at the fluorescent light, suddenly the red cell content would increase 2 to 3-fold instantly. So if indeed something was inside here occupying a lot of space, just by turning on the high intensity light, I must have killed it and make all that volume available for the red cells. So again, this was a key observation, because if you look at the same capillary, just proximal to the site of damage, the red cell content would still be low, and few red cells at a great distance from each other. And as soon as they hit this area that was damaged because of the high intensity light, suddenly the red cells would just widen, occupying the whole diameter of the capillary tube. They would slow down dramatically. So compared to the velocity of the same cells just proximal to this area, they would slow down by 50%. the distance between the red cells would become much smaller, so many more red cells would fit in the same segment. And then the same amount of blood that entered this area, when it left that area, again, the red cells would narrow, become at a much greater distance, and speed up again. Again, showing that the functional diameter of the capillary inside the damaged area was actually increased, and blood could actually use the whole volume of the capillary this side. So just two examples of how that looks in the real images. So this is the segment that I studied in a small capillary just before turning on the light. So again, you see there's only 2 red blood cells into this capillary segment. At that time, we started realizing, looking at higher magnifications, that the red cells were not touching the inner surface of the capillary wall. So there's a little gap between the edge of the red cells and the luminal surface of the endothelium. This is the fluorescent dye labeled column of proteins flowing with the red cells in the center of the vessel. And also measuring the width of that fluid column, we also realized that the width of the flowing fluid column was also significantly smaller than the inner anatomical diameter from wall to wall in this capillary before turning on the light. Well, the same segment is shown here again on the right-hand side. So this is only after 1 or 2 minutes turning on the high intensity light. And then we noticed that just turning on the high intensity light with the dye inside a vessel, suddenly now in the same segment, 4 red blood cells fit into the same vessel, although the width of the vessel didn't change at all. So still the same diameter vessel. And also the width of the dye labeled plasma column suddenly increased significantly to fill the whole anatomical diameter of the vessel. So just by turning on the light, we removed something that was inside the vessel that occupied more than 50% of the volume, because after removing that mysterious compartment, suddenly the blood volume doubled, and also the width of the plasma column significantly increased, also doubling the volume of the fluid plasma. Well, another example, just to give you an idea of the volume aspects, is in a little bit larger vessel. So this, instead of a small capillary with a diameter of 5 microns, this is a small arteriole. You're feeding the capillaries with a diameter of about 12 microns, allowing more than 1 red cell to flow next to each other through the vessel. You see the red cell column flowing. And with these larger vessels, it becomes much more difficult to see the edge of the vessel. And many people realize that you can see all kinds of black lines near the red cell column. It's very hard to tell where the vessel wall actually starts. So if we freeze this frame, so at least you can get a good idea on the width of the red cell column in this vessel. And another method to estimate the true diameter of the vessel, so where the endothelial cells are, is use experimental systems in small animals that are born with a dye inside their endothelial cells. So just looking at the presence of the dye, you can tell where's the real wall of these vessels. So if this is the functional diameter occupied by the blood flowing through the vessel, the next slide shows you where the true diameter is of the vessel. And that's here. So it almost looks like this vessel is much, much bigger than we're looking at different magnification or something. But this is the same magnification of the same vessel, showing the true diameter, anatomical diameter of this vessel. And again, if you compare that with the width of the red cell column, see that the red cell column is much, much smaller than the true inner diameter of this vessel. So another way to see the potential width of the red cell column when glycocalyx is damaged or compressed is to be patient and wait for a large inflammatory leukocyte cell to come along. So these larger cells, they are very low in percentage in the blood. So after a long time, you can actually hope to see one leukocyte to come by. And they are so big and so stiff, they transiently compress glycocalyx and make glycocalyx volume very small for a short period of time, only a few milliseconds. But after, if you are carefully and look at what happens behind the leukocyte, you can actually get a quick peek on how wide the red cell column can be if glycocalyx is compressed. And that's shown in the next slide. So if you pay attention to the width of the column right now when glycocalyx is still intact, If you compress glycocalyx, the width of this column significantly increased. So at least it doubles in width. And the actual diameter of the vessel didn't change at all. So the only thing that changed on the top right, you see the transparent area of the leukocyte. So trailing the leukocyte, just shortly after glycocalyx is compressed, the red cell column width increases by more than doubles. And the volume of the blood inside the vessel again goes by the square of the diameter. So a doubling of the width of the red cell column actually means that the volume of blood inside this vessel increased more than fourfold. So by just compressing glycocalyx transiently, there's 4 times more blood into the same vessel, again stressing the enormous implications of this glycocalyx gel occupying a lot of the volume in the microvasculature and providing a very powerful tool to control blood volume by modulating glycocalyx volume competing with red cell volume. Well, I showed you this already. So now we know that measuring the width of the red cell column in a healthy capillary tells you, how thick glycocalyx is. And by measuring the radial motion of red cells trying to get close to the endothelium, it can tell you something about the dimension and the mechanical properties of glycocalyx. And then another way of looking at that, we also could inject small dyes that actually are small enough to enter into that glycocalyx space. And again, so this gives you a positive image using dyes, again, showing that in a 5-micron capillary, there's a huge amount of volume near the wall of those capillaries occupied by this glycocalyx gel. And again, using electron microscopy, after many years, the person making these images, it's called Bernard Vandenberg. Well, it took him more than 3 years to take these images. And the reason why it's so difficult to see these structures even with an electron microscope is although these structures seem to be very expensive, occupying a lot of the volume inside this glycocalyx matrix, actually the solid structures of the polysaccharide only occupy about 1 or 2% of the total volume of this space. And the other 99% of the volume is actually proteins and water binding to these polysaccharides. And all other people trying to make these pictures. So the first step of preparing your tissues before you can take an image with an electron microscope is to dehydrate your samples, actually, which means that you take all the water out of your samples before you make a picture. So if you take the water out of this matrix, if you don't take care of these structures before you remove the water, all these structures will just collapse into a nanometer scale, small condensed coating on the luminal surface of the endothelium. So it took a long time to think of ways to stabilize these structures and give them enough contrast to see them before people started believing anything was there. So we're now early 2000s. So after 15 years of imaging and developing new techniques, now finally we had a tool to visualize glycocalyx, and people started believing that glycocalyx was indeed something real, occupying a lot of space. And now we and many others could start studying its functional implications. So this cartoon actually summarizes many of the functional implications of that glycocalyx matrix. So on the left-hand side, it's a healthy vessel, the thick glycocalyx represented by the blue polysaccharide structures. And again, as discussed before, this thick coating protects the vessel wall and prevents all other cells in the blood flowing through the vessels to get close to the endothelium. So red blood cells, but also blood platelets for coagulation and leukocytes, so the inflammatory cells, all of these cells never get very close to the endothelium as long as glycocalyx is healthy and thick and protecting the vessel wall. So making sure the endothelium is not damaged and cells do not stick to the endothelium. And on top of that, protective function as a mechanical buffer, we also realized though those polysaccharides of glycocalyx are a very important part of the permeability barrier of the vessel wall, which actually means that the openings between the polysaccharides are so small that individual plasma proteins like albumin or even larger molecules like cholesterol, they cannot leak through glycocalyx. They cannot get close to the endothelium. So the glycocalyx prevents the leakage of plasma proteins and the leakage of cholesterol into the vessel wall, and thereby also prevents the leakage of fluid, because the fluid will want to stay inside the vessel when the protein concentration in the vessel is high. You start losing fluid when you start losing proteins to the outside of the vessel, and then the fluid goes with the proteins, causing tissue edema. Well, in addition to the permeability barrier, we also realized that these structures are crucial for the endothelium to sense whether the blood is flowing slow or fast. So if the blood starts flowing fast, these structures bend forward, and they actually amplify the force of the flowing blood about 100-fold, and then start telling the endothelium that they start to produce nitric oxide. And we know that nitric oxide is a very important molecule that keeps your vessels healthy. And the way that the endothelial cells produce nitric oxide is because of glycocalyx sensing the flow motion of the fluid on top and then telling the endothelium to start producing nitric oxide. While on the right-hand side, you can imagine what happens when glycocalyx is damaged. And now we know that glycocalyx can be damaged by many risk factors that you might be familiar with. So aging is a risk factor, smoking, obesity, diabetes, high glucose levels, hypertension, or genetic predisposition. Some people just have an easily damaged glycocalyx. So when glycocalyx is damaged, well, you start losing your proteins and fluids. Circulating cell like platelets can easily stick to your endothelium and cause all kinds of clotting problems. And also the inflammatory cells, leukocytes, can now easily stick to the wall and causing all kinds of inflammations in your tissues and in your vascular system. And at the same time, nitric oxide synthesis goes down dramatically because the endothelial cells no longer sense the fluid motion of the blood inside the vessel, and there's no trigger for them to start producing nitric oxide. I'm going to show you a few real examples of the science studies that led to all these functional implications that I've shown you in this cartoon. So one of the examples is in large vessels, where we know that at specific sites in large vessels, we have a high risk of developing atherosclerosis, so plaque and lesions in the larger vessels that can cause blockages. So if we zoom in at the specific sites in large vessels where the cholesterol plaques start developing, we realize that these sites where that happens are sites with a very thin glycocalyx. So these sites are very vulnerable and highly leaky compared to the rest of the vasculature that has a very thick glycocalyx. And the reason that these sites specifically start out already at early stages with a very thin glycocalyx is that these sites, if you look at them where they are relative to the large vessel bifurcations, branching points, so these sites where the glycocalyx is very thin, these sites are exposed to very complex flow profiles of blood flow. So this shows how blood is flowing over the bifurcation over a heart cycle. And the red color shows that blood is moving forward in most of your vasculature, except close to these bifurcation areas. If you see a branch coming off at this angle, you see that blood is not just flowing forward, it starts circulating and the profiles become very complex. And there's a small part of your vascular wall and a few endothelial cells where the blood is never moving forward. It's always stagnant or moving backwards a little bit, as shown by the bluish color. And we realize that these endothelial cells, because they never see blood moving forward, they also produce less glycocalyx. And that's why this site is very vulnerable and there's very small glycocalyxes compared to the rest of your vascular system. And again, if we look what happens at this site that has a very small glycocalyx, and one of the functional implications would be a high permeability. It's very, very easy for large molecules like cholesterol to start leaking from the blood into the vessel wall. But first, I'm going to show you some of the tools we use to study the relationship between the level of blood flow flowing over the endothelium and level of glycocalyx production. So the next slide shows you a small device. It's a small chamber that you could open, and on the inside you see this area here. This area we can culture endothelial cells. So the same cells that line the inside of your vessels, we can make them grow inside this little chamber. And we put the lid on the chamber, and we connect on the inlet and outlet. We can connect tubes that have blood flowing over the endothelium, and then start measuring how much glycocalyx is being produced by the endothelial cells in response to different levels of blood flow. This graph shows you that at a normal level of blood flow, there's also a normal level of glycocalyx production. But if flow slows down, or at those bifurcation sites where there's no flow at all, because the blood is stagnant and not moving, there's very little production of glycocalyx at these sites where the blood is not flowing all the time over the endothelium. Well, as I mentioned, one of the consequences is leakiness. So here we studied the leaking of cholesterol into the vessel wall. So at the site prior to the bifurcation area, so the large vessel feeding the two branches, it's called the common carotid in one of the vessels that takes the blood to your brain. And there's a very thick glycocalyx. And this image shows you a cross-section again of the vessel wall. On the top would be where the blood is flowing. The blue colors represent the endothelial cell. And the green dye is the cholesterol that's labeled with the green dye. So you see in a healthy vessel with a thick glycocalyx, all the cholesterol stays confined to the surface of the vessel wall and doesn't enter into the vessel wall and doesn't get close to the endothelial cells. And that's the area where glycocalyx is sitting on the inner surface of the endothelial cells. Now, in contrast, a little bit more downstream at that bifurcation area where the blood is recirculating, and there's very small glycocalyx on those few endothelial cells. Well, within 15 minutes after injecting the cholesterol into the blood, you see not only is it at the surface of the vessel wall, but also now in between the endothelial cells, and a lot of the cholesterol is already inside the vessel wall, accumulating in what we call the subendothelial compartment. So now a lot of cholesterol starts accumulating inside a vessel wall, triggering an inflammatory response, and that ultimately will lead to a cholesterol lesion and plaque formation and blockage of your larger vessels, and then that's called atherosclerosis. Well, another functional consequence that I showed you in the cartoon is the production of nitric oxide by your vessel endothelial cells. So in these studies, we measured the level of nitric oxide production shown on the vertical axis in the vessels with a thick glycocalyx and in vessels with a very thin glycocalyx. So in vessels with a thick glycocalyx, on the horizontal axis, we show the level of blood flow. So if blood flow increases in a vessel with a thick glycocalyx, well, glycocalyx bends forward and tells the endothelium to produce more nitric oxide. So more blood flow, more nitric oxide production. While in contrast, if you have a damaged glycocalyx, if blood flow increases, the endothelium is unable to tell that. It doesn't know to start producing more nitric oxide. While that's supported also by some other experiments by other people, again, over time, this is an experiment where they measured the increase in nitric oxide production in healthy endothelial cells exposed to blood flow. And if they take off glycocalyx, which is the little cross here, if you take off glycocalyx, there's hardly any increase in nitric oxide compared to no flow. Again, demonstrating that glycocalyx is essential for endothelial cells to sense blood flow and to produce nitric oxide. Well, another example is shown in this study. Here you look at a smaller magnification from above to the vessel wall surface. And you see these little dots here, and each little dot is one endothelial cell. So if you start with a situation without flow, and you look at the orientation of the endothelial cells, they look all different directions. So there's no alignment of the endothelial cells. Now, if you have a healthy glycocalyx, you start exposing these endothelial cells to flow from left to right, and you wait 24 hours. Now, by the use of their glycocalyx, these endothelial cells know that blood is flowing from left to right. And as a result, they all start aligning in the flow direction. So they sense the flow, and the orientation of the endothelial cells becomes all in line with the flow direction. Now, if you take off the glycocalyx, as shown in the next slide. After damaging glycocalyx, you see you start out with a random orientation of all the individual endothelial cells. And also after 24 hours of flow, you see there's no effect of the blood flow on the orientation of the endothelial cells. Again, demonstrating that these endothelial cells need glycocalyx to feel blood flow and to change their behavior. Well, another example of the importance of glycocalyx is shown in this slide. This is showing again a cross-section of the healthy capillary on the left and a capillary with a damaged glycocalyx on the right-hand side. So here you see glycocalyx is removed, and as a result, you see a lot of this white space now on the outside of the capillaries. The white space is actually fluid and proteins that leaked out of these capillaries as soon as glycocalyx was damaged. So glycocalyx can be damaged by exposure to inflammatory challenges or different enzymes or smoking. As soon as your glycocalyx is really damaged, the permeability, the leakiness of your small vessels increases, and you start losing a lot of fluid which accumulates on the outside and makes your fingers swell and your ankles swell. So a lot of water retention is due to leakiness of your small blood vessels as a result of glycocalyx damage. When that happens, not only a lot of the volume outside with fluid increases and compresses the small vessels, but also the surface of the endothelium now becomes very sticky and all the blood starts sticking to the wall. So if you count the number of blood perfused microvessels, as soon as you damage glycocalyx, so this is the functional capillary density, so the numbers of capillaries blood perfused, you start out at 100%. Within an hour after really damaging your glycocalyx, for instance, in a situation of bacterial infections like in sepsis, you can lose up to 40 to 50% of all your capillaries when your glycocalyx is damaged and you start losing fluid and all the cells start sticking to the wall. Another example of the functional consequence of a damaged glycocalyx is shown in this study. This is showing the flow of leukocytes, or inflammatory cells, in small vessels. So on the left-hand side, we impaled the larger venule with a small micropipette, and every 5 seconds or so, we inject a small amount of fluid, flushing out the blood. You see in a healthy vessel, those big, large, transparent cells are the inflammatory cells. They usually move very slowly over the vessel wall, just checking if the vessel wall is still healthy, whether it's damaged or not. And if they think there is damage, they stop rolling, and they stop moving, and then they stick with the full body to the endothelium. So the rolling they can do, because they have these pointy fingers that stick out, they can puncture little holes in glycocalyx to scan the surface of the endothelium. But for firm adhesion, they need to make full body contact, which they cannot do when glycocalyx is still in between the column where the blood is flowing and where the endothelial surface is. Well, as soon as we damage glycocalyx, this is just 2 examples showing different methods of damaging glycocalyx. On the left, at the dotted time area, we injected high concentrations of oxidized LDL cholesterol, which is the bad cholesterol that causes vessel damage. Now, when we do inject that, you see within 30 minutes, the number of sticking leukocytes as a sign of inflammation increases significantly compared to a healthy vessel with the healthy glycocalyx. And similarly, if we inject specific enzymes like heparinase that break down specific elements within glycocalyx, also within 30 minutes, you see that many leukocytes now stick to the endothelium, and that the endothelium becomes very sticky when glycocalyx is damaged. Now, at these stages, already early 2000s, we started some early experiments to see if we could fix the glycocalyx and see if the level of leukocyte adhesion would decrease. So we started injecting dye-labeled glycocalyx polysaccharides and see that they would stick to the wall after you induce damage. And if you rebuild a new glycocalyx, so the light compartment inside the vessel wall is now a newly rebuilt glycocalyx. So the black line again is the endothelium. Here's the lumen of the larger vessel where the blood is flowing. So after injecting glycocalyx-like polysaccharides, the same sort of polysaccharides that now we know are also in seaweed and that we use for endocalyx. So if you inject those in a vessel that's damaged, you can see that the new injected polysaccharides or the new seaweeds actually stick to the endothelium and rebuild a new glycocalyx of a few microns in dimension. And again, if you measure the numbers of leukocytes sticking to the endothelium, you see that after injecting the labeled polysaccharides, we had a 50% drop in the number of leukocytes sticking to the wall. And actually, the 50% drop of leukocytes sticking to the wall after repair of glycocalyx was actually due to 100% repair on only one side of the vessel. So to illustrate that, what happened if we injected the therapeutic inside the vessels, the therapeutic was flowing with the blood and didn't reach the other side of the vessel. It only treated the left side of the vessel. So on the treated side of the vessel, we had 100% repair of glycocalyx and 100% preventing leukocytes sticking to the wall. And on the other side of the same vessel, the therapeutic didn't get to this side, and we still had normal levels of leukocytes sticking to the other side of the vessel wall. So the 50% improvement was actually due to 100% normalization on one side and nothing on the other side that wasn't treated. Well, one more example of the importance of glycocalyx is in kidneys. So this is a small kidney unit, a filtration unit where the blood is filtered to take out all the toxins. And the kidney filtration unit is also called a glomerulus. So glomerulus filtration unit actually consists of a lot of small capillaries. So the blood enters the kidney, the small capillaries are perfused inside the glomerulus. Blood is filtered over the wall of the capillaries into the urinary compartment on the outside, and then the clean blood flows out of the kidney again. So if you zoom in on one of these capillaries inside the kidney filtration unit, it looks like this. So this again is a cross section of one of those capillaries, and on the outside would be the urinary compartment where the filtrated toxins would go. And in the dotted square, you see a closer look of the vessel wall, and that's shown on the right-hand side. So this again is the endothelium of a small vessel in the kidney. On the luminal side is all kinds of glycocalyx structures, and on the outside are structures of the urinary compartment. So schematically, it looks like this. So this is the endothelium lining the capillaries in the kidney. And we know that this endothelium in those small capillaries in the kidney is a very special type of endothelium with big holes between different parts of the endothelium, allowing a lot of fluid to be filtered and cleaned in the kidney, because that needs to be done. We need to clear and filter a lot of fluid to get all the toxins out. But people started realizing that these openings in the endothelium in the small kidney capillaries are not real openings. They are filled with glycocalyx. So on top of the endothelium and in between the openings, there's a lot of glycocalyx. And actually the glycocalyx on these endothelial cells and in these openings is crucial to keep all the proteins inside the blood, so that you don't lose your proteins in the urine. These are actually pictures on the right-hand side. So here's the endothelium, and you see these black areas inside the endothelium. So these black areas are areas of highly concentrated glycocalyx filling those openings in the endothelium. So these openings are filled with glycocalyx in a healthy control vessel. So here again is the blood flowing. Here's a lot of glycocalyx filling the openings in the endothelium. And this is the urinary compartment where in a healthy vessel, no protein can be found because it's all stopped by glycocalyx. Now, if you damage glycocalyx, and these experiments use an enzyme called hyaluronidase. Now, when glycocalyx is gone, now you see that there's a lot of transparent openings in the endothelial lining. So the black areas are gone, so glycocalyx is removed. And now if you do that, if you look at what happens with the protein, now you look at the urinary compartment, you see these little black dots. So these black dots are dye-labeled proteins. So in a healthy vessel, these proteins would never go to the urinary compartment and you would never start losing your protein. And you know, if you go to your physician and have your urine checked, they always check for albumin or protein in your urine. So these protein molecules will show up at some point in your urine when glycocalyx is damaged and the endothelium of your kidney capillaries becomes highly permeable. And that's why we know if you do find albumin or protein in your urine sample, that is a sign that your vascular system is compromised, that your endothelium is leaky, and that your glycocalyx is damaged. And that's why high levels of protein in your urine is such a good predictor of future cardiovascular complications. That's because your glycocalyx is damaged. I'm going to skip this. This is just showing that, again, if you measure glycocalyx damage in kidney patients, there's an increase in glycocalyx damage because of a lot of toxins in the blood when the kidney is damaged. But if you do a successful kidney transplant and the kidney is functional again and cleans your blood, you see that the level of glycocalyx in the rest of your body also normalizes. Well, one more example, looking at blood vessels in the eye of people with diabetes. So to look at the health of the microvasculature and the health of glycocalyx, we actually infused different tracers of small molecules going into the whole vessel, penetrating into glycocalyx, and larger tracers not being able to penetrate into glycocalyx. And that gives us a tool to estimate the thickness of glycocalyx. And you see, in people with diabetes, also in the vessels in the eye, glycocalyx is significantly damaged, and that's causing a lot of leakage of proteins and fluids in the microvessels in the eye, and that causes blindness in people with diabetes because of microvascular damage. Now, with that, I would like to stop and give the word back to Bob or Sherry.
Hey, well, thank you very much, Hans. That was very enlightening. I hope that our friends who are watching today learned even more about the importance of, number one, the endothelial glycocalyx, and number two, the role of the microcirculation, and the real miracle of the importance of the blood exchange to support all the organs in the body. So real quick, I wanted to introduce Dr. Mark DeBrincat. Dr. DeBrincat has had the good fortune of having a GlycoCheck unit in his practice now for a little over a month, and has been working with his patients closely, and has also had the benefit. I just realized my camera's not on. There we go. Has had the benefit of offering the MyBodyRx product line to his patients. So I wanted just to get his comments and feedback real quick for a couple of minutes, how Sherry's gathering your questions. So if you have questions, go ahead and submit them, and we'll try to answer as many as we can in the time that we have left. So Dr. Mark, take it.
Hey, thank you, Bob, and thank you, Dr. Vink. I learn so much every time I hear you talk, and I've seen these slides before, but I always take away a little bit more understanding and a little more pearls about how I can turn around and then share that same wisdom, with my doctors and with all my patients as well too. And so I will say, yes, I feel so blessed and honored to be able to have the GlycoCheck system here. And we've had so many patients lining up to get scanned. This is the most dynamic, comprehensive look at the true potential of what my cardiovascular system is actually doing right now. We've, go to doctors and get stress tests and get all these fancy tests on our heart and our big vessels. And, they can, obviously help us understand if it's getting bad, how to avoid a heart attack, but nobody's really talking about the function of our vessels. So with this scan, we can actually see how well they're actually functioning, how much blood's actually going through each one of those size vessels, and how healthy is that glycocalyx is another big one, as well as recruitment capacity and the ability for us to actually be utilizing our blood and getting it to the end point where we need it to be. So it's one thing just to have this great information and this technology now that takes away all doubt and gives us the ability to understand how serious I need to take my health and how hard I need to push to get my life into an optimal level. I mean, I think that's pretty much what people want, at least what my patients want, is they want to be not just out of pain or past this little crisis, they want to be optimally better. And when it comes to cardiovascular, the only advice we're getting is, hey, eat clean and exercise. You know, that's what's so important, cardio in water and so forth. And we do all these things, but yet heart disease is still the #1 killer that gets everybody. So clearly we're not getting the full picture. And now all of a sudden we have the ability to get a full picture, to know exactly what my markers are. But more so than that, we have the good news of a solution, of an actual product that we can now take to grow that glycocalyx back. And when we do that, you realize this is our largest organ in our entire body. And when we can help the function of our largest organ work, life is in the blood. And if blood can get to the tissue, we're going to remain living and bring life into this tissue and pull the stuff out. Like Dr. Hans was saying, that when the glycocalyx is bad in the kidneys, we're losing stuff out of our blood that we shouldn't be losing. And we're leaking out our proteins, and then we're going to eventually starve our organs if we're not getting blood through those smallest vessels. And so organs will fail. And so this is a way that we're not only helping prevent a lot of things, but we're going to save a lot of lives. There's a lot of people that are getting really close to the end with all sorts of very chronic illnesses that don't really have a solution to get the drains moving and to bring the blood into the areas that they can't get it. They don't have instruments. I guess until now, we have an instrument that's so small that it can penetrate a microvessel. But you think about people doing surgery on bigger vessels, there's just too many of the small vessels, and that's where all the magic happens. So we have a product that's been patented to actually grow these fibers back that allow them to start functioning normal again. And so obviously we're super excited about this. The world absolutely needs this technology, and we're so excited that you're finally bringing it out. I mean, we love the fact that you've done so much research over the last decade, 3 decades, and have gotten it to this point that we can launch a product like this and give it to the world and really have all of everything in place for it to absolutely fit perfectly and flow nice. People want the research, they want the science. Of course, the patents are bonuses that, you know, gives us the security that nobody else is going to make that same product and be able to have that same claim. We really are an amazing company, and they've created an amazing piece of technology, and then the supplements that bring us the good news of how we're going to heal and how we're going to actually heal from the inside with things that matter. Supplements that are our building blocks to build those glycocalyx back in the areas that are damaged. And so we'll learn a lot more about that later. But, you know, thanks for letting me A, have the unit, and then B, be able to speak up and be able to share the good news of how exciting this is. And the world is ready for it. We're sick and tired of being sick and tired. And to be able to have such an amazing product to turn our health around so quickly is awesome. So thanks again for both of you for all you've done to bring us to this point, and looking forward to see how far it's going in the future.
Well, thank you very much, Dr. DeBrincat. This has been our life mission, and we're excited to share this knowledge with all of you.